The overall goal of this method is to direct the differentiation of human pluripotent stem cells toward the primitive or definitive hematopoietic program using a stage-specific signal manipulation approach.
This method can address many key questions in the field, such as understanding the signal and transcriptional regulators of human definitive hematopoiesis. The main advantage of this technique is that it uses stage-specific manipulation of Wnt signaling. Demonstrating this procedure is Carissa Dege, a postdoc in my laboratory.
To begin the protocol, obtain previously prepared human pluripotent stem cell lines, or hPSCs, grown on mouse embryonic fibroblasts, or MEFs, to 70 to 80% confluency in a 37 degrees Celsius incubator with 5% CO2. Aspirate the hPSC media and add 1 milliliter of 37 degrees Celsius 0.25% trypsin EDTA to each well for 1 minute. After 1 minute, aspirate the trypsin and add 1 milliliter of stop media to each well. Using a cell scraper, scrape the cells off the plate. Then, add 1 milliliter of wash buffer to each well. With a 2 milliliter serological pipette, triturate the cells 3 to 5 times to break up large clumps. And transfer the cells to a 50 milliliter conical tube. Centrifuge the hPSCs at 330 times G for 5 minutes.